Appreciate the invitation, and it's good to be amongst friends here talking about how to advance biotechnology in agriculture. I'm going to talk to you today about the work we're doing in aquaculture. Anna just gave a great overview of aquaculture, so that will save some time for me, and thank you for that. As we get started here, as Chantal mentioned, our company's mission is to commercialize genetic technologies to really improve aquaculture productivity and sustainability. And a big focus of that is around genome editing and how to get that technology into commercial use. As a company, we have genotyping, genomics, breeding, and genome editing teams, and all are really needed as you move genome editing toward commercialization. We work with the industry in various aspects amongst these four channels. And a big focus for us, as Anna just mentioned, is sterility. There's the basic piece, which is biocontainment is likely needed in aquaculture to implement biotechnologies, and it's a great benefit there. There's also environmental stewardship. There's all these problems with escapes and permitting and industry expansion, and it does give performance benefits in some species that mature during the grow-out cycle. So it's been a major focus of our genome editing research over the past 6 years. And I'll talk a little bit about where we sit today. But first of all, genome editing is just, as we all understand, another tool in the toolbox for improving genetic performance, ranging from very simple mass selections through more sophisticated using genomic selection. So genome editing will be an adjunct to this. We won't get rid of traditional breeding, but we'll be able to enhance its productivity and the rate of improvement. Escapes of improved fish are a major issue. And as Anna just said, it's unusual that there's all these constraints around testing animals when genetically improved animals are farmed all the time, and they do escape. And so in salmon in particular, it's become a political, a social, and economic issue in the mainstream news. And this is an example of why sterility is so important in aquaculture. The fish that do escape, so massive numbers can escape. 160,000 fish escaped in Washington from Atlantic salmon. These are all Atlantic salmon. In Chile and Norway, it's tracked and reported, and large numbers of fish can escape. The impact of that is a whole study of science and environmental science, and the gene flow from improved species back to the wild populations is a reasonable concern, especially when you look at the numbers. This is an estimate on my part from looking through the literature, but the numbers of salmon, farm salmon in Norway is around 150 million fish. The wild population is around 600,000, somewhere in that area. And the numbers of escapees varies per year, but you can have as many as 200,000. So there is probably significant pressure from escaped fish on the wild population, both in Norway, and then there's numbers there on a global scale, like salmon farming globally dwarfs the wild population. And so the escapee pressure can be significant even before we start talking about biotechnologies. So a sterility solution is reasonable to pursue, especially if we're going to do additional modifications and put the fish into the environment. And this issue, while debatable on the impact on wild species, has become politicized as a way to damage the aquaculture industry or prevent expansion of the industry. So NGOs can take this topic, probably inflate the risk a bit, and use it to damage and create regulatory barriers. And as we probably all, a recent announcement in Canada, for example, is that in British Columbia, they're going to force all salmon pens out of the water by 2030. So there's some really strict problems coming that impact our ability to feed people. And so solutions to these that help us farm and really a lot of it's politics too, but an environmentally sustainable manner are needed. So other examples of this is cichlid populations and invasive potential tilapia in Africa is a concern. And serial breeding programs in Hawaii are limited to stocking F1 species, F1s for a breeding program only. So you can't really advance the production of the fish, and you can only farm native species there. And then many aquaculture regions are restricted to stocking native species and then restricting the amount of genetic improvement that you can add to those species. So really all of the understanding that we have in agriculture on genetic improvement can be constrained in how we deliver production efficiency and the ability to feed people in a sustainable manner using genetics. So we really need a solution to this problem. So as we all have discussed and are familiar with here, we have a conventional breeding program where we have generation intervals and genetic gain over time. Genome editing for certain traits gives us the ability to dramatically increase the speed of genetic improvement. So we can make major increases in value created for producers, for consumers, and for environmental sustainability. And what I'm talking about today, and when I'm talking about genome editing, is the variety that we're using is using CRISPR or CRISPR-associated technologies, creating new variation in a gene or knocking out a gene, and there's no new DNA added. So it's the least objectionable form of biotechnology that we can think of. And there's lots of applications for genome editing in aquaculture. Anna did a great job going through this, but even to circle around, it's not just increasing productivity, but you can increase the nutritional benefit and decrease the environmental footprint of the industry. And this is true for agriculture in general. In particular, for aquaculture, there's more of a focus on the environmental component, as we just discussed. But you can look at everything from waste output, how much feed the fish use, the flesh quality, the nutritional benefit, the production characteristics, resistance to disease, and sterility. Those are all areas of active research. So in the end, our conclusion has been that most farmed genome-edited aquatic species should be 100% sterile. So there's an exception there if the fish are otherwise biocontained, so they're farmed on land or in other forms of containment, sterility is probably not required. But because all aquaculture species are still relatively wild, we haven't had the 3,000 years of domestication that other species have had. And much of fish farming takes place within the wild environment. Like 99% of production are in ponds or net pens or lakes. There's always a possibility that scapies could outcompete or interbreed with wild counterparts to a genetic deleterious outcome. So it's irresponsible to add major genetic changes like GE without sterility. So the work Anna's doing is very important in this regard. So on sterility side, biocontainment is a key. 100% sterility is a direct route there, and we believe we need to get to 100% sterility. It should ease the regulatory burden for genome editing and subsequent biotechnologies for fish. So then it opens the door for other applications, like using transgenics or the aqua bounty line of salmon in larger applications. It also allows commercialization of genetic improvement, so it protects investment in genetics. Aquaculture industry is pretty well known for the exchange of genetics in some species. People develop them, and then it's pretty easy to collect the fish and commandeer the genetics for yourself. It also improves performance and grow out for many species. And so the one caveat there, as I said, animals raised in RAS environments, so that's on land, may not require sterility because they are otherwise contained. So from our perspective, we regard sterility as a fundamental requirement for the commercial application of genome editing in farmed aquatic species. So there are, as I said, benefits to sterility in addition to the more practical environmental and regulatory benefits. So you get, in many species, a better growth rate because the fish aren't maturing and diverting energy to maturation. They're growing better. You get uniformity in size, better feed conversion, behavioral aspects that can take place during sexual maturation, such as aggression or territorial behaviors, can be reduced, and the economic viability of farming can be increased. So there's some practical reasons to pursue sterility as well. So just summarizing some of the existing concerns with aquaculture. Aquaculture scapies may create genetic contamination. They can establish feral populations. You can potentially produce a decline in biodiversity. There are, as we've discussed, some legitimate concerns here. Much of it's overblown and been politicized, but there is a base level of reasonable concern. So effective containment prevents farm species escape. It minimizes impact on the environment and native species. So if you have sterile fish, you prevent reproduction, maintain ecosystem balance with escapes. Native populations don't have to, there's no concern of feral populations of tilapia colonizing areas, and leads to more sustainable aquaculture practices. So talk a little about the work we're doing. So we have a commercial solution at this point that we're going to market with this year. So we'll have 4 licensees working on commercializing this. So we have a genome editing process that produces 100% sterile fish for aquaculture. So there's I think a pathway here forward. So it is commercial scale at this point. So it's technically, I'll go through this. This is more scientific. I'm happy to answer questions on this and go into more detail if there are questions. But basically, we can take advantage of the facet of fish, which is really interesting, is that you can do germ cell transfer from one fish to another, and the germ cells will still mature as normal. So if you take a donor testis, extract the spermatogonial germ cells and transplant them into a sterile recipient, those germ cells will colonize the gonad. So you can see the red cells here have colonized the recipient, and they will produce germ cells. So new sperm or eggs. So you can see there in the fish that have been transplanted, normal ovaries and testes develop, and in the fish that were sterile and not transplanted, those fish developed as sterile fish. So this is really interesting because you can have fish parents that have progeny that are unrelated to them. So if you extract fish from the germ cells from the orange fish on the left and implant them into donor recipients on the right, you have a sort of this turquoise colored fish with orange fish gonads, and they produce progeny that are from the orange fish lineage. So this opens up some interesting applications, both in propagation of species, shortening generation time. This actually works between species in some cases. So, you know, a rainbow trout could have salmon progeny. So there's a lot of things you can do, but from our perspective, we use this as a tool for sterility, and that you can create somatic mutations that inhibit or prevent germ cell development or sexual development, and transfer the germ cells from those fish into sterile recipients. Those recipients then function as normal broodstock on commercial scale and produce 100% sterile progeny that you can then use for stocking in sea cages. And because you can test the genes, this process with appropriate quality control generates 100% sterile progeny that you can then be confident that you're farming 100% sterile fish. So this works quite well. As I said, it's commercial scale, and then it's durable as well. So we've done a lot of this work in tilapia and testing the ideas. The genes that we're working on are conserved across species, so it should work exactly the same in all species. We're testing in other species now, but the genes that create sterility really are sterile. So you can, reproduce tens of thousands of fish at this point. You can monitor them over long periods of time, 12 months, 14 months. I think we're up to 16 or 18 months now in tilapia, and you see no signs of germ cell development over that entire period. So it's working quite well on a commercial scale. As I said, there are some benefits. Just having all male tilapia gives you, sterile tilapia gives you a 12% increase in growth over traditional males if they're sterile. So you do get some benefit from this environment, this energy conservation. But also we're doing other work on performance genes. So we've tested these genes in tilapia as well. So you can create edits, and there's 3 genes we're working with. And you can see you can get some really interesting phenotypic benefits in just a single generation with the edit. You get up to a 68% increase in growth, 12% better use of feed, so better improvement in feed efficiency. And then gene 3 is the myostatin gene most people we're working with, and you still see some growth in feed efficiency benefits, but in that one you get a major benefit in fillet yield. So there are, in addition to sterility, production traits that are ready to go and be commercialized, and seem quite straightforward. As Anna mentioned, these have all been tested in the laboratory, so none yet have been tested on a commercial scale. So we still have that last step to go, but we're ready to take that step. So the way we think about this is sterility plus. So we're going to go create sterile fish anyway. You might as well add these other performance traits at the same time, and don't duplicate the work. And so you can get a line of fish that are sterile and have really significant improved performance benefits that should streamline the regulatory approval process. There are already some regions where the process is straightforward. So as Anna mentioned, Japan would be an example. Brazil is an example as well. But in other areas, even in Japan, even though it's straightforward, they can't put the fish in sea cages yet because the government would like to see some sort of containment solution. So maybe there's a solution there for Japan, but it also becomes then this platform for future genome editing or other biotechnology applications to aquaculture. And so even if you just model this out, we just did a model of the Brazilian salmon industry. If you implemented this across that industry, which is about 450,000 metric tons of production annually, and then it's increasing. A simple model shows you add about $260 million worth of value to the farmers and processors. And you also remove some of the concerns with feral tilapia populations escaping. So it's really has the potential to be a sizable impact, and also can increase the amount of food we can produce. We're trying to feed people a nutritious diet. So there's a lot of upside here on the technology. We're really excited about the potential and that gets ready for commercial scale application. So with that, I'll just summarize sterility. 100% sterility is feasible with the genome editing approach. This is going to help address concerns around farming and performance in aquaculture. And I think the important part here is this is the foundational technology on which to build biotechnologies for aquatic applications. It solves a major concern of regulators and stakeholders. So it seems like a good place to start. With that, I'll thank the team at CAT. So this is our scientific team here, led by Xavier Lau, and really strong team. They've done a lot of great work. It's been a pleasure to work with them over these years. And with that, I'll say thank you for your attention and happy to take questions later.